The overall goal of this experiment is to describe how bacteriophages are synthesized from their genome using an all E. coli cell-free transcription-translation system.
This method can help answer key questions in the synthetic biology field, such as elucidating the molecular self-assembly mechanisms in living systems.
The main advantage of this technique is the versatility of the platform. Many biological systems can be fully recapitulated and finely tuned for experimental analysis.
Begin this procedure with preparation of phage plaques as described in the text protocol. Use the end of a sterile Pasteur pipette to core and remove a single plaque. Blow the plaque into the diluted cells. Then incubate at 37 degrees Celsius for 2 hours. To test for full infection, take a 500 microliter sample into a 1.5 milliliter tube and add 10 microliters of chloroform. Immediately vortex the mixture at high speed and observe if the solution clarifies, which indicates phage infection. Once the cells are fully infected, incubate for another 2 hours. Next, add DNase before centrifuging the cells at 8,000 times G and 4 degrees Celsius. Discard the supernatant and resuspend the pellet in 10 milliliters of 1x tris magnesium chloride. Add 500 microliters of chloroform and vortex at high speed to lyse the cells. Clarify by centrifugation at 12